So in today's lesson, I wanted to do a quick overview of some of the details related to DNA replication that I felt the video didn't delve into in enough detail, and to outline some common misconceptions that students have typically. So I want you to take a quick look at what I have constructed here in my diagram already, and try to make sense of what's going on here. What I'm hoping you're noticing is that at this point we have our two strands of our double helix that have begun to separate. So DNA helicase has done its job already, and presumably we have some SSBs, some single-stranded binding proteins, keeping these two strands separated. And what that creates for us is a replication fork. And so here we have the beginning of where replication is going to happen. And one of these is presumably going to be our leading strand, and one is presumably going to be our lagging strand. I want you to first off take a look at the labels that I have on my two strands. I have a 3' and I have a 5'. I have a 5' here and a 3' here. Why is it that these seem to be inverted? Well, I'm hoping that you know the answer to that, which is that DNA strands, the two sides of the DNA ladder, that is, are always positioned anti-parallel to one another. So although it's really difficult to show in this diagram, I've simply labeled to indicate that by using the 5' and 3' for directionality. So now I want you to pause the video, and I want you to think about which of these two strands, and I suppose I could just call this strand A and then call this strand B. I want you to think about which of these strands, A or B, would represent the lagging strand and which would represent the leading strand, and why. I want you to pause the video now and think about that, and come back when you're ready and you've written down some sort of a response. Okay, well, this is the point that I was hoping to outline for you, where students often have a misconception. So we know that no matter what, DNA is constructed in the 5' to 3' direction, right? And so often students will say, okay, well then 5' to 3', this is going to be continuous, and therefore this A is going to be discontinuous. But that's in fact incorrect, because we need to remember that the new strand that's going to be built, so let's just say I start a strand here, that new strand is going to have to be anti-parallel to the existing strand. And so what that means is that in my new green strand here, what I've started with is my 5', and it's going to continue to be constructed in the 5' to 3' direction. So in fact, the end part is my 3'. So what does that mean? Well, that means that this construction is going to continue on and on and on and on without interruption as my replication fork opens. So in fact, A is my leading strand for that reason. Now, strand B, on the other hand, in my new strand, it also is going to be anti-parallel, and so that means that anti-parallel to strand B, that is. So this will be my 5' direction in the new strand, and here's my 3' end in the new strand. So it's going to have to be constructed discontinuously, because of course, what would have to happen is I'd have once the fork opens a little bit, I can construct this little bit. So 5' to 3' is going to get constructed. Then my little fork opens up a little bit more. I can lay out a new RNA primer and build a new little section of DNA, again in the 5' to 3' direction, and then again. And so what that results in are a bunch of these discontinuous sections just waiting for that fork. And of course, our fork right here would continue to open, and then as it opens, then I am able to build a new little fragment in the 5 to 3 direction. And so each of these fragments is referred to as an Okazaki fragment, and we call it a lagging strand, of course, because in each case I would need to set up a new RNA primer, my new landing dock for my DNA nucleotides. And that tends to slow things down a bit, right? And at the end of all of this, these fragments are discontinuous and need to be glued together using DNA ligase to create a complete new strand. So that's it. I hope that made sense. Please reach out if it doesn't make sense to you and if you're unsure. Just remember, DNA is always built in the 5 to 3 direction. We say it's built in that direction, and so that means that any new strand will have to be anti-parallel to the existing parent strand. And that affects how we designate the leading and the lagging strand.